Good morning, everybody. My name is Cornelia. I'm the product manager for gene function and gene editing at Takara Bio Europe. So I hope that the sound is fine and you can hear me. So you will be muted during this webinar, but you can type your questions into the questions box at any time, and we will have time to discuss those at the end. So today I'm very pleased to present you our novel CRISPR-Cas9 Gesicle technology and also our tools for AAV delivery to improve your gene editing experiments. This is just the outline of the talk today. So after a brief introduction, I would like to show you our CRISPR-Cas Gesicle production system, explain the workflow and how it works. I will then show you some data showing Gesicle-mediated gene editing in different cell types. First of all, for knockout of an exogenous Z green cassette, and then data for the knockout of the endogenous CD81 gene. I will then talk also about off-target effects and the advantages that Gesicle delivery has with regards to off-target effects. And in the second part of this talk, I would like to introduce you to our AAV Pro CRISPR-Cas systems, which are AAV serotype 2, and I will present the features of the AAV Pro SP Cas9 system and the SA Cas9 system. So this is just an outline of a typical CRISPR-Cas gene editing workflow, and the first step is usually the guide RNA design. This is a very important step, and what you would like to get is really a guide RNA with high efficacy and also with high specificity and low off-target effects. So there are many in silico tools nowadays. I would also like to invite you to visit our website under clonetech.com/guide. We have a list of available tools and also a video with tips on guide RNA design. So as all these in silico tools have or use different algorithms, I would recommend also maybe to use two different tools to choose your guide RNA, because maybe a guide RNA which is best in one tool will not be the best one with another tool. In-house, we also tend to design 4 to 5 guides per target gene or per target site. And we also test our guide RNAs in vitro using our guide RNA screening system prior to the delivery into cells. The next step is then, of course, the delivery into your target cells. And again, here you need to choose a method with which you can obtain efficient delivery, but also very low toxicity, and this may depend on the cell type you're using. And of course, at the end, you would like to get efficient editing with no additional footprint in your target cells. You can then quickly check editing efficiency in your cell population. There are different tools available on the market for this quick screening of cell populations. We also have a tool called the Mutation Detection Kit, and I will show you some results using this kit later in this talk. And then if you find that the gene editing efficiency in your population is good, you can go for the single cell cloning step, which is a quite tedious step to isolate your single clones. And here, of course, the goal is to maximize the cell survival, because usually cells do not like to be isolated from the cell population. And then at the end, you can screen your single clones and identify those that carry the desired mutation, and also check for specificity of the gene editing event and for off-target effects. And then finally, you will isolate and expand your edited single cells. So today I would like to focus on the delivery part of this workflow. And as you may know, there are many different ways to deliver Cas9 and also the guide RNA. For example, both can be delivered using lentiviral vectors. This can be important, for example, for guide RNA library screenings. AAV is another viral option, and I will talk about this option later in this talk. Plasmid transfection can be used in easily transfectable cells, but also direct delivery of mRNA of Cas9 together with synthetic guide RNAs or in vitro transcribed guide RNAs. And finally, Cas9 can also be delivered as protein, for example using protein transfection or electroporation, and also as so-called RNP complex, so a ribonucleoprotein complex, meaning that the Cas9 protein is already complexed with your target-specific guide RNA. And Gesicles are a novel technology to deliver such Cas9 RNP complexes. So now you're maybe wondering where the name Gesicles comes from. So Gesicles comes from nanovesicles and glycoprotein because Gesicles are such vesicles which are made in a producer cell line after overexpression of a specific glycoprotein. So as you can see here on this picture, Gesicles can be used to deliver cargos, and in this case, for example, a protein like Cas9 or an RNP complex such as Cas9 complexed with a target-specific guide RNA. And as Gesicles also express VSVG on their surface, they can be delivered to a similar range of cells as would be expected for VSVG pseudotype lentivirus, meaning that the tropism of Gesicles is very broad mammalian. So now our CRISPR-Cas Gesicle production system is it's a bit do-it-yourself system in which you can produce your own Gesicles that will contain both the Cas9 protein and your target-specific guide RNA complex to the protein. So let's have a look at the workflow of Gesicle production, which is quite similar to lentiviral packaging. And you will use a so-called producer cell line, which is based on 293 T cells. And you will just do a big co-transfection of these producer cells. And you will co-transfect your target-specific guide RNA plasmid together with a so-called Gesicle packaging mix, which is provided in our kit. And this packaging mix contains, so it's a lyophilized mix, and it contains already the extract transfection reagent. And then several plasmids, plasmids encoding the nanovesicle inducing glycoprotein, plasmid encoding Cas9 endonuclease, and the plasmid encoding the cherry picker red fluorescent protein. So now the overexpression of the glycoprotein will then induce the Gesicle budding from the producer cells. We also use a second clone-tech technology in the system, which is called iDimerase. And iDimerase is a system to artificially induce protein-protein interactions. And we just use this technology here to tether the Cas9 protein to the membrane-bound cherry picker fluorescent protein. And this is just used to enrich the Cas9 at the membrane and to enrich Cas9 in the Gesicles, because without this the system, the Cas9 due to the NLase would go into the nucleus and would not be enriched in the Gesicles. The loaded Gesicles will then just pinch off the producer cells and can be collected from medium. This is also very similar to lentiviral packaging. And what's also nice is that you can store the Gesicles for later usage at -70 for over 1 year. So you can also make a bigger Gesicle production and then just freeze the Gesicles for later usage. When you apply the Gesicles to the target cells, they will just fuse with the target cell membrane and then release the Cas9 protein together with your guide RNA to the target cells for gene editing. So here is again in brief the workflow. All you need to do is to clone your guide RNA into the provided plasmid and add this plasmid to the tubes with the packaging mixes, and then add the mix 1 to the mix 2 and do this big co-transfection of the 293T producer cell line. Then you will also need to treat the producer cells with a so-called dimerizer molecule. Remember, this is just to tether the Cas9 to the membrane-bound cherry picker protein to enrich Cas9 within the Gesicles. And then you will wait 2 to 3 days and you can collect the Gesicles from the supernatant, and there's a centrifugation step to concentrate those. And then you have your population of ready-to-use Gesicles. So now when you apply the Gesicles, the target cells will also get red thanks to the cherry picker fluorescent protein. So this is a nice control to check whether delivery was efficient. And you can also use this red fluorescence to cell sort the positive cells, so to enrich your positive population. However, the red fluorescence will not tell you whether you really efficiently packed the Cas9 into your Gesicles. You could also deliver just empty Gesicles and your target cells would get red with a cherry picker. So to check for efficient Cas9 packaging, I would just recommend a simple Western blot. So you can lyse a fraction of your Gesicle production, and then, for example, use our Cas9 antibody to check for the presence of Cas9 in the Gesicles. What you can see here compared to recombinant Cas9, that our Gesicles, or in this particular production, contained about 30 to 50 nanogram of Cas9 per microliter of Gesicles. And you can also see that this Cas9 here migrates a little bit higher compared to the recombinant one because it carries a little tag for the dimerization. Another way to check for the presence of Cas9 is, of course, to check your target cells after delivery. And again, you can use our Cas9 antibody for Cas9 labeling or immunostaining. Here's just an example in RPE cells where you can see Cas9 which is present after Gesicle delivery. You can also see the red fluorescence here of the cherry picker protein. So to label for Cas9 after Gesicle delivery, you need to do this quite quickly because remember, you will deliver the active protein. And it has been shown that Cas9 protein is rapidly degraded after protein delivery. So I would expect that almost all Cas9 is degraded within 24 hours. So here the staining has been done 12 hours later. This also means that here you cannot see any effect on the knockout yet because 12 hours later it's a bit early to see any effect on the fluorescence of this ZS green cassette which has been targeted. Okay, so now I would like to show you some results using Gesicle delivery. So first of all, we created an easy model system to show Gesicle efficiency in different cell types. And for this we created stable cell lines containing the ZS green reporter, and we then designed a guide RNA against ZS green. So ZS green can be easily measured in the flow cytometer, giving a fluorescent readout. And you can imagine by targeting ZS green when a frame shift or a knockout mutation occurs, this should lead to the functional knockout of the protein, and this can be measured by a leftwards shift in the fluorescent readout in the flow cytometer. So we tested Gesicles with a ZS green knockout in various cell types. On the left you can see easy to transfect cell types, and we compared directly to plasmid transfection, which are the gray bars. And on the right you can see harder to transfect cell types. So for the easy to transfect cells, you can see that sometimes plasmid delivery gave higher efficiency for the ZS screen knockout, and sometimes Gesicle delivery gave higher efficiency. This was depending on the cell type. In any case, you will expect lower off-target effects when using Gesicles, even in those easy-to-transfect cell types, and I will speak later about the off-target effects. So this is still one advantage of Gesicle delivery. Now, of course, the more interesting cell types are the hard-to-transfect ones. Here you can see that plasmid delivery was really very inefficient for the gene editing or inexistent, and with Gesicle delivery we could achieve correct gene editing efficiencies of 20 to 40% in those cell types. The only cell line which was quite resistant to gene editing was the KBM7 cell line, which is the leukemia cell line. So of course we also wanted to knock out an endogenous gene, and we choose the CD81 surface receptor, and here are some results again in Jurkat cells, which are rather difficult to transfect, for example. And again we compared directly to plasmid transfection. The readout is again fluorescence because we labeled this surface receptor with a FITC conjugated antibody, and then you can just measure the intensity of fluorescence to check for protein expression. So you can see here that the plasmid delivery again was not very efficient. There's still a big population showing the CD81 expression, and then only 7% knockout. In contrast, Gesicle delivery showed a nice functional knockout of the CD81 protein, and you can see here 76% of functional knockout. You may also have noticed that here the efficiency in the Jurkat cells is much higher compared to the ZS screen knockout. This is due to the fact that the CD81 usually it's just one copy present in the cell, whereas the ZS screen cassette and the stable cell lines, there can be multiple copies of the ZS screen, so it's more difficult to knock out. So yeah, again, very nice efficiencies in Jurkat cells for the CD81 knockouts. We also wanted to test Gesicle delivery to human iPS cells, and we just used the standard protocol here with a bit longer incubations. So we tried two different incubation times of Gesicles, 6 hours or 24 hours. And then the CD81 levels were measured 7 days later. And you can see here that we obtained very good results also in the human iPS cells with 40 to almost 50% of functional knockouts. For the gene editing in iPS cells, we used our Cellatis DevCS culture system, which is a complete system for culturing, also containing the matrix. And it's really a great system because it also allows single-cell passaging and single-cell cloning. Interestingly, Gesicle delivery had did not show any toxicity and had no effect on the pluripotency for the markers we tested. So the last point I would like to discuss with regards to Gesicles is off-target editing. So it has been published by others that Cas9 protein or Cas9 RNP delivery can help to decrease off-target effects. And this is explained by this very short burst in Cas9 activity. The protein will be active very quickly, but will also be degraded very rapidly. So we wanted to check whether this is also true for the Gesicles. And to assess off-target editing, we used our guided mutation detection kit. So this is the tool I mentioned that can be used also for very quick screening of your cell population. And this technology is sometimes also called the mismatch detection assay, and I would just like to explain how it works first before showing the results regarding the off-target effects. So as I said, this kit is very good for a quick screening of cell populations because after gene editing you will end up with a mixed population. Some cells will carry different mutations and some cells will still be wild type. So the first step of this kit is to amplify the genomic DNA of the target region, and this kit here uses Terra polymerase, which is a very nice enzyme to do direct PCR. So you do not need to purify your genomic DNA. You can start with a crude extract. And as you are starting with a mixed population of cells, you will also get a mixed population of DNA or PCR fragments, some carrying different mutations and some will be wild type. And the enzyme, the guided resolvase, which is included in our kit, will specifically cleave mismatched DNA. So you will just denature and re-anneal your DNA, and this will scramble all the molecules, and some mutated molecules will anneal with the right type, and this will create mismatches. So those mismatches will be specifically cleaved by the resolvase, and you can then run this on an agarose gel, and the ratio of uncleaved versus cleaved fragments will give you an estimation of the mutation frequency in your cell population. Okay, so we used our guided mutation detection kit to assess off-target effects and compared again Gesicle delivery to plasmid delivery. And we choose as target gene the EMX1 gene. And you can also see that there's a potential off-target sequence in the genome which is only different or which differs only by 3 mismatches to, compared to the EMX1 site. Now, when analyzing the plasmid delivery and with a mutation detection kit, you can see here that we had about 16% of gene editing events at the EMX1 site, as shown here by the cleaved fragments. But there was also a high frequency of gene editing events at the off-target site. Now, in contrast, when using Gesicle delivery, you can see similar efficiency at the EMX1 site, but the off-target site here was really very clean and comparable to the negative control. We then wanted to go one step further and used our guided indel identification kit, which is just a complete system to clone your edited sequences and to send this to sequencing. So here we isolated really single clones and sequenced them. And again, we compared the plasmid delivery here on top to the Gesicle delivery. On the left hand, you can see the EMX1 target site. You can see here the different editing events. And on the right, the off-target. And as you can see here, for the plasmid delivery, again, all the clones we sequenced carried also gene editing events at the off-target site, whereas after Gesicle delivery, all the clones were really wild type. So this confirmed that what has been published before, that RNP delivery can help to reduce off-target effects. So to summarize this first part, I hope that I could show you that Gesicles effectively deliver Cas9 guide RNA RNP complexes to a broad range of cell types, and also including difficult to transfect and non-dividing cells, and also iPS cells, for example, as I showed. So the major advantage is the Cas9 protein delivery, because there's no Cas9 coding sequence present, so there's no chance for genomic integration and no chance for persistent nuclease expression, and there's also no elevated overexpression of the Cas9 nuclease, which can be observed, for example, when using plasmid delivery, and all this together helps to reduce the off-target effects. So I would like to spend the second part of this talk to show you another tool that we provide, which are our AAV Pro CRISPR-Cas systems. So again, it's another tool for the delivery step of the workflow. And a bit similar to also to Gesicles, AAV delivery can help for genome editing in hard to transfect mammalian cells. And AAV can also target proliferating and non-proliferating cells. AAV is also suitable for in vivo delivery. It's a virus which has a very low intrinsic immunogenicity, and our systems produce AAV2 serotype, and this serotype targets muscle, liver and brain in vivo. Another interesting feature regarding AAV, in contrast to lenti or retrovirus, is that AAV is a non-integrating virus. So this means AAV will just allow for transient expression of Cas9 and the guide RNA, and this is also helping in reducing the off-target effects. So now the AAV Pro CRISPR-Cas systems we offer, so we have two different systems. The first one is the CRISPR-Cas system delivering Cas9 from the Streptococcus pyogenes, also called SP-Cas9. And there's just a little problem with the AAV and the Cas9. So as you may know, AAV is a quite small virus, and the problem is that Cas9 is a very big has a very big coding sequence. It's a very big protein. So this large size cannot be packed into one single AAV virus. And that's why in our system the Cas9 coding sequence is split between two AAV vectors. So as you may know, Cas9, the SP-Cas9 uses the PAM sequence NGG, which is very frequent in the genome. Our second system is called the SA-Cas9 system, and so using the smaller Cas9 from the Staphylococcus aureus. And what's very nice with this Cas9 is that you can pack it into one single AAV vector, which makes the system much easier to use. The SA-Cas9 does not use the same PAM as the SP-Cas9, so the PAM is longer, it's NNGRRT, where N is any base, and R here, the R's can be an A or a G, and the T is preferred but not strictly required. So before going into more detail with each of these systems, I have some general remarks using our AAV Pro systems. So first of all these systems are complete systems. They also contain the ligation reagents to clone your guide RNA into the provided vectors. And our CRISPR-Cas systems also contain the packaging vectors. So our AAV Pro systems use helper-free packaging, which increases the safety. So meaning you do not need an adenovirus or herpes virus to produce your AAV. One of the packaging vectors also overexpresses a specific microRNA, and this doubles the virus titer. This is shown in this figure here. The overexpression of this microRNA in the packaging cells really greatly helps to increase the viral titer compared to a packaging vector without the guide RNA, the microRNA. And the last point I would like to mention is that our systems also contain the AAV extraction solution. And this solution really gives you also higher titers compared to the classical freeze-thaw, which is shown here in this figure. We have shown extraction of different AAV serotypes, and each time you can see here and that the extraction solution in blue gives really higher titers compared to the freeze-thaw method. So this is just to give you an overview of all the components which are included in the CRISPR-Cas system. So now let's have a closer look at the SP-Cas9 system. So as I said, it's a split system because the large size of Cas9 cannot be packaged into one single AAV virus. So now how does it work? What you will get are two different vectors, the guided up vector containing an upstream portion of Cas9 and the guided down vector containing a downstream portion of Cas9, and both portions have 1.6 kb homologous overlap. The down vector is also provided pre-linearized, so all you need to do is just to ligate your little guide RNA sequence in. And then what happens is that you will produce AAV particles for the up and the down vector, and you will co-transduce your target cells with equal MOI of both viral particles. And in the target cells there will be homologous recombination thanks to this overlap, and this will create full-length Cas9 and also together with your guide RNA, and all these will be expressed in your target cells. So you will just produce two separate AAV particles of the up and the down, and the full-length Cas9 will be created in your target cells. Here are some results compared to plasmid transfection. Again, we used our guided mutation detection kit to assess the mutation frequency in the cell population, and here we targeted the CCR5 receptor. We compared AAV delivery to plasmid delivery, so which is here the P, in 3 different cell lines, HEC293T, so very easy to transfect cells, and then more hard to transfect cell lines, HepG2 and MCF7. So what you can see here, in any case, AAV delivery increased the gene editing efficiency in all these cell types in the HEC cells from 30 to 60%, and here of course it's even more important. HepG2 cells, we could increase the indel percentage to 17%, or here to 25% in MCF7 cells. So this means that AAV is a really good option for hard to transfect cells. So now last year the assay Cas9 has been identified and described here in this Nature paper by Feng Zhang's group, and what these authors found that assay Cas9 could edit the genome with similar or very similar efficiencies compared to SP-Cas9. And what's a very important feature of this assay Cas9 is that it's really 1 kb shorter, which facilitates packaging into AAV vectors. In terms of specificity, this group has also shown by genome-wide off-target editing analysis. So this group, they used the BLESS technique. They showed that assay Cas9 has very high specificity and even a little bit better specificity compared to SP-Cas9. And other groups have then also used so-called GuideSeq to confirm these data. So assay Cas9 has similar efficiencies to SP-Cas9 and very high specificity. And what I said, what's very nice with the assay Cas9 is that it's much smaller compared to the big SP-Cas9, and this allowed us to create one single vector to express assay Cas9 and to clone in also your target-specific guide RNA. We provide this vector pre-linearized, which really facilitates the cloning of your target-specific guide RNA sequence. So all you need to do is just to order a DNA oligo of 20 nucleotides or 22. I think it's a bit longer for assay Cas9, and to clone this into the pre-linearized vector. So now we wanted to compare the efficiency of assay Cas9 and SP-Cas9 also in a side-by-side experiment. And here we choose the exon 1 of the CYP2 gene and designed guide RNAs either for the SP-Cas9 or for the Cas9. So two different guides for this exon, and we were lucky because the PAM sequences could be used here for both enzymes. Okay, so now the results. We analyzed our cell population in 3 different cell types, HEC, MCF7 and HAPG2 cells, again using the mutation detection kit. And what you can see as an overall result is that the overall efficiency of Cas9 in our experiments was also similar to that of Cas9. Meaning you can see here for the Cas9 overall efficiency on these 2 target sites was ranged from 15 to 38%. And for the Cas9 it also ranged from 13 to 42. So we can say that overall efficiency was very similar. But what we also noticed is that Cas9 and Cas9 can have different targeting efficiencies on the same region of the genome. Meaning that, for example, for the Cas9, it always performed better at this first target site here, whereas we had the reverse result for the Cas9, which had always higher editing efficiencies at this site here. So although they have very similar efficiency, they might differ on a very specific target sites. So this brings me to the end. I would like to summarize the second part. As we just saw, AAV transduction is a very efficient method, especially in hard to transfect cells compared to the plasmid delivery. Another interesting point with the AAV is that it's also suitable for in vivo delivery, and as I said, AAV2 targets brain, liver and muscle cells in vivo. AAV is a non-integrating virus, so this means there's no persistent expression of the Cas9 and no risk that it will get somewhere into the genome. And this also helps to reduce off-target effects. Our AAV Pro CRISPR-Cas systems are really complete systems, which also include the helper-free packaging vectors, the vector expressing the microRNA and the extraction solution. So this helps to increase safety and give you high titers. And the Cas9 is a smaller Cas9 autolog with similar efficiencies and very high specificity. And what's very nice with the Cas9 is that you can pack this into one single AAV vector. So thank you very much for your attention. If you have any questions now, please type them into the questions box, or you can also drop me an email later on to our tech support address. And I would also like to invite you to check our website under www.clontech.com/guided. So I will just check whether there are any questions.